Go.
Alrighty, listen up. Welcome to the Genetic Investigation Bureau. I am your lead investigator, Hanalei Nia Vicarpas, and this is my partner, Forensic Analysis, Leon Mel Pastor. We have a new case on our hand. It's a big one. We're not dealing with a typical crime scene. We are dealing with a crime at the most fundamental level of life itself. This is the case of the corrupted code.
That's right. So our job today is to understand the crime, so which is the genetic disease. Okay, so and learn about the most advanced tool in our arsenal to fix it, the gene therapy. Okay, so every one of you in this room acts as the witness. So your observations will be crucial to cracking this case. Okay, so but before we start, um, let us first define the learning outcomes or know the learning outcomes. Okay, so at the end of this lesson, so the learners should be able to define what a gene is, differentiate gene therapy from stem cell therapy, explain the importance of gene therapy in the future, demonstrate processes of gene therapy, and finally show in life situation genes' importance in education.
But first and foremost, let's warm up for a forensic eye. With a series of blurred and cryptic images, there are clues to a larger puzzle. What do you see? The first one is the DNA, or diaronoxic acid. The next one is ribonucleic acid. And the third one is our topic, the gene therapy. To solve this case, we need to understand the basic. Every living thing has a genotype, its original underlying genetic blueprint, and the phenotype, the physical manifestation of the blueprint, which can be influenced by environmental factors. Think of it as a suspect plan and how the plan actually plays out in the real world. But it's not always straightforward. Sometimes the genetic code is tricky. We have phenomena like penetrase. Does everyone with the code corrupted show symptoms and expressivity? If they show symptoms, how severe are they? This is why the same genetic crime can look different in different people. So the first is understanding what is gene therapy. In genetics, the genotype and phenotype are the two ways of describing an organism's traits. The genotype is the genetic code, while the phenotype is the physical expression of the trait. Here is a closer look at what is genotype and phenotype with an example. Genotype, genetic code responsible for a trait, while phenotype is observable traits and characteristics. For the examples, genes of color is a genotype, and the phenotype is brown, green, and blue. How about the heritable? Yes, it's inheritable, and while in phenotype, it's not necessarily. How about to identify the genotype? DNA analysis, family history, or by the test crossing. While in the phenotype, it's the simple observation by the eyes. And then lastly, genotype depends on genetic code, while in phenotype, genetic code plus environmental factors and epigenetics. Factors that determines how one's genotype correlates to one's phenotype. The first one is penetrase. What do you mean by penetrase? Penetrase is the proportion of individuality with a specific genotype who also express the corresponding phenotype or trait. In cases of complete penetrase, every individual with genotype will show the trait, while in incomplete or reduced penetrase, only a portion of areas will develop the trait. Expressivity is the degree of which trait expression differs among individuals, and like penetrase, expressivity describes individual variability, not statistically variability among the population of genotype. And the third, this is an example of expressivity. Individuals with the same genotype can also have different degree of the same phenotype. Example of this feature is Marfan syndrome. So this is how it starts. health symptoms, such as being tall, thin with long, slender fingers, where others also experience life-threatening complications involving the heart and blood vessels. So this picture shows Marfan syndrome, where you can see the fingers is in different position and also the body, chest, and how the person looks like. This is also an example of Marfan syndrome. Another example. So you can differentiate what is Marfan syndrome to an ordinary or normal individual. Next will be the genetic mosaicism. Mosaicism, also known as genetic mosaicism, is a phenomenon in which a single multicellular creature has more than one genetic line as a result of genetic mutation. This suggests that a single fertilized egg resulted in many genetic lineage. For example, the color of the eyes of the boy explains what is genetic mosaicism. So this is what you call chimerism. So it spells like in which occurs with two or more genotypes appear in one individual in a similar way as mosaicism, is also commonly mistaken for genetic disease. This is what you call also hyperpigmentation, the darkness of the skin. And the next one is two different color eyes, here seen in the picture. And the red blood cells, or the DNA, which have two DNA in one body. The next will be the X inactivation. X inactivation refers to an irreversible inactivation of one of the X chromosome in each cell of a female during early embryonic development, resulting in inactive X becoming heavily methylated and adopting a heterochromatic configuration. So disease that results due to inactivation is X-linked diseases and gen expression.
Okay, so now moving on to the gene therapy. Okay, so what is a gene therapy? So a gene therapy involves altering the genes inside your body cells in an effort to treat or stop the disease. So basically, the genetic or this gene therapy is like a solution for the genetic problems or the genetic diseases. Okay, so this gene therapy is a medical treatment that uses genes to stop or prevent diseases. Okay, so an example for this is in a form of inherited blindness called the liver congenital amaurosis. Okay, so this liver congenital amaurosis patient who are legally blind can now see after a single injection of a correct gene directly into the retina. So this isn't science fiction, so this is happening now. Okay, so moving on to the genetic engineering. So this genetic engineering involves the removal of a desirable gene from a gene, adding it to the gene structure of a bacterial cell, replacing the recombinant DNA into the bacterial cell. So the bacteria then have the capability to produce the protein by the original animal or plant cell. Okay, so to further understand, let me state an example. So in medical field, so let's say the synthetic insulin. So this production of synthetic insulin is a classic win for genetic engineering. Okay, why? Instead of relying insulin from pigs, they inserted human gene for insulin into a bacteria. Okay, so this bacteria became tiny factories producing pure human insulin for diabetics worldwide. Okay, so this shows the ability to reprogram organisms to solve medical problems. Okay, so the first synthetic insulin produced using the E. coli bacteria in 1978. So, and was first sold commercially in 1982. So, under the brand name Humulin by ND and Company. Okay, to further understand, magagat sa talo, further example in research. Okay, so scientists are now using a tool called the CRISPR-Cas9. Ganun siya. Spelled as the CRISPR-Cas9. So, CRISPR-Cas9 is a precise gene editing technology that creates genetically modified animal models of human diseases. Okay, so this allows them to study how diseases like Alzheimer's progress and to test new drugs in a controlled environment to help them accelerate their discovery.
So there are two procedures in fixing genes. The first one is germline gene therapy, and the second is somatic gene therapy. The first one is germline gene therapy. Germline gene therapy is defined as the introduction of normal human genes into the egg, sperm, or early embryos of parents with the goal of altering the genetic inheritance of their offspring to prevent genetic diseases or introduce enhancing genetic variation. While the somatic gene therapy inserting forensic gene into somatic cells, which are any cells in the body except sperm and eggs. It is also known as somatic gene therapy. This process involves introducing therapeutic DNA into non-reproductive cells to treat diseases in the individual with the key distinction that any genetic changes are not passed on their offspring. So, common genetic disorder. What do you see in the picture? Analyze the picture. So, the picture is an example of Down syndrome. So, how many chromosomes are in our body? So, there are 46 chromosomes. 23 from the mother and 23 from the father. So, in Down syndrome, the 21 chromosome is in there is one mutation of one gene, which means there are 3 in a one cell. So, Down syndrome is a genetic condition where a person is born with an extra chromosomes. This can affect how their brain and body develop. People diagnosed with Down syndrome can lead to healthy lives with supportive care. The next one is analyze the picture again. So, there are normal red cells that you can see here. This is a normal red blood cells, and this is our blood vessels. This stiff and sticky sickle, this is what you call sickle cells. So, sickle cell disease, sickle cell anemia is the most severe type of sickle cell disease. An inherited blood disorder. In sickle cell anemia, you inherit a genetic mutation change that causes abnormal C-shaped or sickle-shaped red blood cells. So, as you analyze or you see in the picture, our normal red blood cells is circle, while the sickle cell is in the C4. So, what will be gonna happen in our blood cells if there will be a sickle cells? It will become a blood barrier to our blood vessel, which means that the normal red blood cells cannot penetrate Merkel form. It's a rare inherited metabolic disease that results in mental retardation and other neurological problems when treatment is not started within first few weeks of life. So what will be the symptoms of PQ? So about 50% untreated infants have the following early symptoms: vomiting, irritability, eczema, or the unusual odor of skin color, and lastly is the unusual odor of urine nervous system. The fourth one, analyze again. In the first picture, what if you got yourself? This is a normal stages of blood flow. Magawas ang dugo, ma-dry ang sod, which means naa siya'y airway. Well, if hemophilia, magawas ang dugo pero may blockage. Dire, magawas ang dugo pero mag-dry. Pero dire sa hemophilia, dili siya maayog dali, inability to clot or maglisod gawas ang dugo. Hemophilia is a rare disorder in which the blood doesn't clot properly, resulting in the disease's hallmark symptoms of excessive bleeding that extends over a prolonged period of time. That in turn leads to other complications. So which means, maglisod siya kaayo. Ang mga tao nga na hemophilia is dali na siya lang mabunog. Kung makat ang dalilang kamot, dugay maayo, it takes prolong of time. The effects of germs in our environment to be and also be discussed by myself. The first is effect of light or chlorophyll production. The second is effect of temperature on hair color.
Okay, so do not forget what we're ultimately fighting for. So the gene mutation. Okay, so a gene mutation is a permanent change in the DNA sequence that makes up a gene. So these changes can be caused by errors during DNA replication, exposure to environmental factors like UV radiation or other types of DNA damage. Okay, so basically a gene mutation is the root cause of corrupted code we see in this case, or in our case 5. So a gene mutation can also result in different proteins being produced, which may alter organisms' traits or cause diseases. So an example for this is the hereditary breast cancer. So this hereditary breast cancer can be acquired through genes. So, but most cases are not directly caused by inherited genes. These BRCA1 and BRCA2 are common genes associated with hereditary breast cancer. So this BRCA1 and BRCA2 normally help repair damaged DNA. So when mutated, they became less effective and has a higher chance of cancer. Okay, so moving on to the DNA nucleotide subunit. So there are 3 subunits: the phosphate group, deoxyribose, and the nitrogen base or nitrogenous base. So the phosphate group, but before that, let us first define gene. Okay, so we mentioned naman ito earlier in Miller, no? So that gene is composed of subunits called the nucleotides. Okay, so each nucleotide is composed of 3 smaller subunits. Ito, the phosphate, deoxyribose, and the nitrogenous base. Okay, so this phosphate group is a group of atoms that is negatively charged and forms the backbone of the DNA. So, muna sila ang chemical group made up of phosphorus and oxygen. So, another is the deoxyribose. So, they covalently link to phosphate group, also contributing to the backbone. Muna sila ang 5-carbon sugar made up of carbon, oxygen, and hydrogen. So, moving on to the nitrogenous base or the nitrogen base, a chemical unit composed of carbon, oxygen, genes. So, na siya 4 ka bases. The adenine, thymine, cytosine, na kani sa guanine. So, these four bases or four nitrogenous bases are specific sequence for that genetic that encodes genetic information. So these four nitrogenous bases contains instructions for making a protein. So
The next topic will be the gene expression. Gene expression is the process of using information in a gene to create a functional product, such as a protein or an RNA molecule. Gene expression, this is tightly regulated process that occurs in two main steps. The transcription, where the DNA sequence is copied into messenger RNA or mRNA molecule, and translation, where the mRNA is used to assemble a chain of amino acid into a protein. Then regulate of a gene expression controls when, where, and how much of a particular product is made. So, the picture shows the process of gene expression. The transcription, where it copied the gene. Next one is the mRNA. So, RNA means ribonucleic acid, and lastly is the translation, where the protein is made or the amino acid. DNA code. This code is organized into three basic units called codons, which determine the sequence of amino acid used to build a protein. The DNA code is similar to a language, where the order of the bases forming words are codons that specify the building blocks of life. The genetic code, heredity. So we all know what is heredity. We have a further knowledge about it. So heredity is the passage of this instruction from one generation to another. For offspring to resemble their parents, it requires a set of genetic code for specifying its traits. Genetic engineering, a technique of controlled manipulation of genes to change the genetic makeup of cells and more genes across the species boundaries. So, welcome to the Q&A time. So, the team will gather the evidence. Now it's time to test our knowledge. We're going to spin a question related to see which aspect of the case will review. No backing down, students. So, okay, we will gonna choose 10 students to answer, and we will gonna interview what they have learned about it. Excellent work, team. You've connected the dots. Remember, the genetic code doesn't operate in a vacuum. Environmental factors can influence how genes are expressed, adding another layer to our investigation.
And never forget what we're ultimately fighting. So the gene mutation. So these permanent changes in DNA sequence are the root cause of the corrupted code we see in our case files.
So through the powerful technique of genetic engineering, we have the potential to not just solve these cases, but to prevent them entirely. This concludes our briefing on the case of corrupted code. The evidence is clear, the tools are powerful, and the future is in our hands. The mystery of genetic disease is being unveiled. One gene at a time, you are now deputized against of the GIB disease. Thank you, and that's all.